Now that we've looked at the digestion of proteins into amino acids, let's focus on what can happen to those amino acids within the body. So we've mentioned the carbon within the amino acids can be used and stored in the form of glucose or triglycerides. Alternatively, it can be oxidized in the TCA cycle to generate energy. The nitrogen from the amino acids is primarily used in the urea cycle. And the urea cycle is a means of detoxifying ammonium ions and incorporating them into urea, which is excreted in the kidney. Ammonium ions are the most prevalent form within the body, as the pKa is 9.3, such that the protonated form is the most common molecule. However, keep in mind, free ammonia can also be found, and within the kidney, free ammonia is essential for disposal of protons, such that ammonia plus protons in the kidney will allow for the excretion of ammonium ions. So we're going to start by looking at the process of starting with amino acids and leading to the release of ammonium ions. The next part of this will focus on how ammonium is incorporated into the urea molecule. So it's important to note that the urea cycle is unlike many of the pathways or all of the pathways we've looked at to date. The pathways we've looked at, such as glycogenolysis or gluconeogenesis, are specifically active in either the fed or the fasted state. In the case of the urea cycle, it can be activated in both the fed and the fasting state. And the activity of the urea cycle is really a function of excess amino acids within the body. So if we start with dietary protein, which we know is digested to free amino acids, if these are in excess, the freed ammonium ion is used in the urea cycle, while the carbon can go to glucose or triacylglycerol synthesis, or the amino acids could go to protein synthesis. So keep in mind that in the fed state, if there is an excess of amino acids, these amino acids are deaminated and the ammonium ion will be funneled into the urea cycle. Similarly, in the fasting state, gluconeogenesis is occurring. And the primary substrate for gluconeogenesis is amino acids, specifically alanine from the skeletal muscle. The movement of these amino acids into the liver to be used as a substrate for glucose production requires, again, the amino acids to be deaminated, and that ammonium ion is used to generate urea. So in both the fed and the fasted state, the urea cycle plays a role in either the disposal of excess amino acids or the deamination of amino acids as a means of providing substrate for gluconeogenesis. So the movement of the ammonia from amino acids happens using transamination reactions. And these transamination reactions require the cofactor PLP, or peroxidyl phosphate. And we've seen this reaction before in Module 6. Essentially, it starts with a single amino acid and a keto acid, usually alpha-ketoglutarate. And through the transamination, the amino acid donates its ammonia to the keto acid, and this results in an alternative amino acid and its conjugate keto acid. If we look at this example below, in the case of aspartate reacts with alpha-ketoglutarate, and the movement of the ammonia to the alpha-ketoglutarate from the aspartate generates the new amino acid, glutamate, and the keto acid of aspartate, which is oxaloacetate. These reactions are readily reversible, such that they're involved in both the degradation as well as the synthesis of amino acids. So there are several different pathways that funnel into the generation of ammonium. First off, both glutamine and asparagine contain multiple amides. You should review the structure of these amino acids. In this case, glutaminase, or asparaginase, will catalyze a reaction to release either glutamate or aspartate and a free ammonia. This is particularly important in the kidney, where the ammonium produced is excreted directly in the urine, and it forms salts with metabolic acids. Glutamate can be further deaminated through the reaction using the enzyme glutamate dehydrogenase. This enzyme will free an ammonium ion from glutamate to generate the keto acid alpha-ketoglutarate. This reaction is reversible, and in the forward reaction, as shown here, the release of ammonium from glutamate requires the cofactors NAD+ or NADP+. This is one of three reactions we'll look at where ammonium can either be freed from an amino acid or fixed to a keto acid. As illustrated here, there are other amino acids which also can contribute ammonium to the pool of free ammonium. You can see here serine can be deaminated to form pyruvate. Threonine can be deaminated to form alpha-ketobutyrate, and histidine to form urocanate. And all of these reactions can contribute to the pool of free ammonium. You'll also notice the purine nucleotide cycle in both the brain and the muscle. In both the brain and the muscle, ammonium released by amino acids can be scavenged by glutamate. Glutamate will then transfer that nitrogen to oxaloacetate to generate aspartate. The aspartate will then enter into the purine nucleotide cycle, contributing that nitrogen to the generation of the ring structure, and fumarate is released. You'll also notice here that a significant amount of ammonium is generated within the gut. And this is due both to bacterial contributions as well as amino acid digestion. Before moving forward, I want to take a minute to discuss the reaction catalyzed by glutamate dehydrogenase. As mentioned previously, this reaction is reversible and takes place in the mitochondria of most cells. Again, it can incorporate ammonia into glutamate or release ammonium, shown here, to generate alpha-ketoglutarate. This is an important reaction, as this ammonium can then be incorporated to produce urea. In this direction, going from glutamate to alpha-ketoglutarate, again, ammonium is released, and NAD plus or NADP plus is required as a cofactor and is reduced to form NADH or NADPH. This is the primary direction going from glutamate to alpha-ketoglutarate if urea is being produced. This is a mechanism by which ammonium can be removed from the body and carbon skeletons can be generated and funneled into the TCA cycle and used as needed. Alternatively, the reaction can go from alpha-ketoglutarate to glutamate. In this direction, free ammonium is then fixed
2-alpha-ketoglutarate to generate glutamate. In that reaction, NADH or NADPH is oxidized.
So glutamate, again, will play a pivotal role in this pathway for us. And it can be generated through transamination using peroxidyl phosphate or through the reaction we just looked at using glutamate dehydrogenase. Glutamate can then function as an ammonia carrier for amino acid synthesis. Glutamate reacting with alpha keto acids of any number of amino acids can result in the reconstitution of alpha ketoglutarate, which is the keto acid of glutamate, and the amino acid from the keto acid that glutamate reacted with. So again, glutamate is pivotal to the shuttling of ammonium ions into the liver for the urea cycle. This starts with any number of our amino acids, which again could come from the diet or through protein catabolism. These amino acids undergo a transamination reaction with alpha ketoglutarate to generate a keto acid and glutamate. Again, we're just transferring the amide group from the alpha carbon from the amino acid to alpha ketoglutarate to generate glutamate. Glutamate can then be directly deaminated using glutamate dehydrogenase to generate free ammonium, or it can undergo an additional transamination reaction with oxaloacetate to yield aspartate. And aspartate will contribute our second ammonia group in the urea cycle to generate our urea, which contains 2 nitrogens. So it's really this part of the pathway here that's really important and pivotal on the role of glutamate in shuttling free ammonium ions into the urea cycle or to aspartate. So we understand that amino acid degradation and uptake is not just happening in the liver. So there needs to be a mechanism for transporting ammonia in a non-toxic form from peripheral tissues to the liver where the nitrogen can be used to synthesize urea. One of the primary shuttles for doing this is the use of alanine. Alanine can be released from the skeletal muscle. If we think about the case of protein catabolism, where amino acids are required for gluconeogenesis, proteins are degraded in the skeletal muscle to yield amino acids. These amino acids react with alpha ketoglutarate via a transamination reaction to yield glutamate. Glutamate can then aminate pyruvate to generate alanine, and alanine is shuttled to the liver, where, through a series of an additional transamination reactions, alanine can be divided into its nitrogen-containing component and carbon-containing component and can be used to synthesize glucose. Glucose can then be shuttled back to the skeletal muscle. This entire cycle is referred to as the glucose-alanine cycle.
In addition to alanine, glutamine is also used as a non-toxic carrier of ammonia in the body. In muscle, as well as most peripheral tissues, the transport of nitrogen looks like this, where alpha ketoglutarate will fix free ammonium using glutamate dehydrogenase to generate glutamate. This glutamate will be used to synthesize glutamine using glutamine synthetase. And this enzyme will fix another free ammonium onto glutamate, again to generate glutamine. Glutamine is then released from the peripheral tissues and will travel to the liver. In the liver, this glutamine will contribute 2 ammonium groups which can enter into the urea cycle. The first ammonium is released through the actions of glutaminase. This will then generate glutamate and free ammonium. Glutamate can then be used to generate alpha-ketoglutarate by our enzyme glutamate dehydrogenase, freeing another ammonium ion. And again, both of these ammoniums can enter into the urea cycle. The process of nitrogen transport we've just looked at on the several previous slides is summarized here. Again, if we look at nitrogen transport in most peripheral tissues, you can see that glutamate is going to be primarily used to fix free ammonium to generate glutamine. Glutamine will then be released from these tissues and taken up by the liver. Glutamine can be deaminated initially by glutaminase to release glutamate and free ammonium, and the second nitrogen group can be released from glutamate using glutamate dehydrogenase, illustrated here. These two ammonium ions can be used to produce urea and will contribute one of the nitrogens to the synthesis of urea. Keep in mind the other nitrogen will come from aspartate. Alternatively, in the muscle, illustrated here, is the alanine glucose cycle. Again, you have glutamate dehydrogenase fixing ammonium to glutamate, which can be used in a transamination reaction with pyruvate to generate alanine. Alanine can be released from the muscle, enter into the liver, be transaminated back to pyruvate. This pyruvate can be used to synthesize glucose, and again glucose will be released from the liver and can be oxidized in the skeletal muscle.
Before we move on to looking at the urea cycle specifically, let's see if we can deduce when the urea cycle would be most active. And a 25-year-old male fasted for 48 hours. Postprandially, so after a meal, in a 10-year-old who consumes a typical American diet. Postprandially, in a 48-year-old man who consumes a typical American diet. Or postprandially in a 27-year-old pregnant female. First off, let's think about when the urea cycle is most active. We've said it's active in both the fasted and the fed state. The urea cycle is also reliant on the amino acid pool. When amino acids are being shuttled to protein synthesis, there are fewer amino acids to be degraded, such that the urea cycle is less active. This helps us eliminate both B and D. In the case of children, these individuals are growing rapidly and synthesizing any number of proteins. So in this case, regarding the 10-year-old male, although the urea cycle is certainly active, it's not the most active in this scenario, as many of the amino acids he's consuming are being incorporated into new proteins. The same can be said for the 27-year-old pregnant female, where protein synthesis is clearly active in the developing fetus. That leaves us with A and C. In a 25-year-old male fasted for 48 hours, or a 48-year-old male who consumes a typical American diet. Although in the fasted individual the urea cycle will be elevated, after 48 hours we still have sufficient glycogenolysis occurring, such that gluconeogenesis, although it is happening, is not happening at a great extent. That leaves us with C, which is the correct answer. In this individual, where there is little new protein synthesis besides regular protein turnover, and the diet contains ample protein. There will be ample room for amino acid degradation, and the urea cycle will be most active under these conditions.